Good afternoon, everyone. Welcome to the first webinar of 2013. My name is Sue Tappan. I'm a staff scientist here at MBF. I've recently changed my name, so you may know me and be more familiar with my maiden name, Susan Hendricks. I'm joined today by Mike Fay. Mike, introduce yourself.
Hello, everyone. I'm a member of the software development team here at MBF since 2007, and I've done some work on most of the tools that we'll be demonstrating today.
We've joined forces before on a webinar dealing with the Zeiss apotome, and today we're going to be talking to you about brain mapping techniques in Neurolucida. As always, we love hearing from you both during the webinar and after, so please submit your questions via the question window and we'll address it as we go along. This webinar is being recorded and will be available for viewing later. So we've got a lot to get to, so let's get started. Neurolucida, since its very beginning, has had the functionality not only to analyze single complex neuronal trees, but also to map regions and the location of cells for the purpose of brain mapping. Neuroanatomical mapping uses a number of physical sections through the region of interest, either the entire brain specimen or through a subset of the brain, and we're going to show you today how to take data, organize it into serial sections, as well as some techniques for automated contouring and cell detection. Version 11 is soon to be released. Mike, when do you think it's going to be available?
Version 11 is in the late stages of testing right now, and when we're comfortable with how stable it is, we'll be releasing, probably in the very near future.
All right, so we're going to give you a little taste of what you can expect in version 11, and some of the highlights that we're going to show today include how the serial section manager and image organizer controls now make it easier for you to organize your data. There are also major changes to how images and virtual slides are created and used that also improve functionality and throughput for your data collection. So what is brain mapping? It's a methodology that can be utilized to understand the anatomical organization and distribution of cell populations within regions. Brain mapping is used for many reasons: to map a particular circuit or projection pattern using an anterograde or retrograde tracer, identify the distribution of implanted stem cells, or to locate cells using a particular reporter like GFP or that produce a particular protein like calretinin. These are just a few examples. In these cases, physical sections on a glass slide that are at a regular spacing through your region of interest are stained for the population that you're interested in following. Then the slides are either imaged or placed on the microscope and analyzed using Neurolucida. In Neurolucida, you will delineate the anatomical regions at your desired level of accuracy and use markers to plot the location of each individual cell.
So Sue, what do you mean by a desired level of accuracy?
Well, I'm glad you asked. Absolute accuracy comes in two flavors when you're performing brain mapping. The anatomical specificity is actually very important because you want to be certain that the marker that you're placing for the cell that you're interested in is within a particular region of the brain. And so understanding where you are in the brain by using an atlas or consulting somebody is very important. The actual number of markers that are placed is of less importance. We're using this method, this brain mapping, in order to guide more detailed analyses later on. And so in this case, we may be more interested in representing that there are many more cells labeled in layer 2, 3 than in layer 5, for example.
I gotcha.
So if you are interested in an absolute number of these cells in various regions of interest, you may consider using a different technique, stereology, for an unbiased estimate of the total number of your cells in your region of interest. Today's webinar will demonstrate brain mapping techniques using a long-time Neurolucida user's data. Dr. Carton has used our software for over 20 years, and this image data is a component of a research study that is soon to be published in the April issue of the Journal of Comparative Neurology. In this paper, Drs. Guillard, Carton, and Suvet used cholera toxin injections to the unilateral retina to map retinal projections in the Nile rat. We love hearing from our customers. Please tell us your ideas for new tools and mechanisms to improve your research. Dr. Carton has, and I'm pleased to show you what we've changed in Neurolucida to make brain mapping and many other aspects of using Neurolucida and Stereo Investigator better in version 11. So today we're going to create maps of cell populations. We're going to demonstrate how to manage serial section data when you have multiple sections per image file. So if you have a very large virtual slide or an image from a slide scanner, or in the case of a single section per image file.
So we're going to show you a number of tools that we have made available that make it easier to view, edit, and create your data. We'll demonstrate the automatic outlining of regions of interest, which today will be serial sections. We'll show you how to automatically detect cell populations of interest. We'll show you how to efficiently use the serial section manager, and finally how to visualize and navigate the serial section data that you create.
So we're going to use lots of different aspects of Neurolucida in order to create and utilize this data. So here's a little subset of some of the major functions that we're going to be talking about today. Serial Section Manager is going to be heavily used, as is Image Organizer. In addition, I'm going to show you a nifty technique where you can combine Where Is mode and Edit mode to very quickly and easily adjust the locations of your markers and contours.
We're going to show you how to automatically detect objects you're interested in and outline them. We'll show you how to automatically detect objects and place markers on them. And we'll also show you how to do auto-contouring, which can be done live. So while what we're demonstrating today can be done live on a microscope system, we'll be working with virtual slides. And for those that are unfamiliar with the term, virtual slides are images containing multiple fields of view that have been stitched together to enable large areas of interest to be stored in one digital image. The images we're using today are comprised of hundreds, if not thousands, of individual fields of view, which means we're using gigabytes worth of pixel data. So virtual slides enable users to share information easily, as well as work away from the microscope when necessary.
Great. So for our first demonstration today, we're going to show you how to create models using virtual slides in which the image contains multiple sections. We're going to map the visual circuit from these image sections that have been labeled with cholera toxin subunit B. We'll then organize those sections according to the rostral caudal order and then show you how you can view and inspect the detection. So let's get to it. We're going to switch to Neurolucida. And so one of the first things that I've done for our demonstration today is streamline my version of Neurolucida. So I've removed toolbar buttons that don't pertain to brain mapping, and I've organized all of this by creating a profile specific to the task that we're doing today for brain mapping. So you can have multiple profiles that represent either different hardware configurations or even different tasks that you're going to be doing. You can also set this up according to individual lab members as well, if you would prefer. And so for the brain mapping, you can see that we have no icons that relate to tracing neurons because that's not what we're doing today. In addition, I have a number of windows that have been docked. I have the macro view available here. Below it, I have the serial section manager. And then I also have the image organizer. So I'm going to start by opening a data file that I've created in advance to get us started. So I'm going to zoom out. You can see in the macro view that I have 3 individual image files loaded, and each of those individual image files has 8 histological sections. And so now I'm just adjusting our view by using the icons to zoom in and zoom out. And so you can see that in the image organizer I have 3 files in the image organizer, and you can see that I have 3 images available in my tracing window. So our first task for brain mapping is to delineate each individual section so that we can then probe those sections to look for labeled neurons.
Okay, I'm going to show you how to use the automatic object detection to quickly outline your serial sections. This application, things tend to work best when you're zoomed out a little bit. So the tool that we're talking about right now is under the image menu. You go to automatic object detection and you select outline detected objects. So the way this tool works is by growing clusters of pixels of similar color, representing detected objects ultimately. So by adjusting parameters for the detections, in most cases it is possible to get high quality results that are reproducible by saving them down here. This little button has a file cabinet icon, and you can save different parameter sets that work on different types of tissue. So the detection can either work on a single full image at a time, or you can use regions of interest. So you can trace contours, and if you're looking for cells inside those contours, we'll only run the detection around where those regions of interest are. You can define the object color simply by clicking on what you're looking for, or you can use a color picker to define a color. Ahead of this, we already played with parameters, so we could get a parameter set that we know works well, and we've saved it as section outline. So when you select it, it runs the detection and shows you a preview. Here the outline is in green of what's going to happen when you finally run the detection and we'll make real contours. So how's that look, Sue?
Oh, it looks pretty good.
So I'm going to say outline my objects.
Oh, that's so much faster than me manually tracing each of those 8 sections.
That was pretty quick. So now we can do this again for another image. We select the image we want to run.
In the image organizer.
We go back, open our detection tool, select our preset again, shows what we're going to detect, and say outline, and then one more.
And so now the outline is in red.
Outline my objects, and now we just traced 24 sections really quickly.
Wow. Okay, well I think the detection is pretty good. I like those contours, but some of those contours have some points that I don't think that should be there. So let me show you some ways to quickly edit your contours. So I'm going to zoom in a little bit more so that you can see things in greater detail. And if we look at this section here on this lower right, so the last section in the lower right, you can see that the contour comes down and around the cerebellum, and I prefer if it didn't do that. So you can see here that it's a little too tight for my liking. So I'm going to go into Where Is mode, and when you select Where Is mode, which is this icon here with a question mark, you can see that you can see all of the individual contours and tracing data for the entire data file in one fell swoop. I'm going to combine that with Edit mode, so I'm going to go into Edit, select Objects, and now I'm going to select the individual points, checkbox here. Now, if I draw a marquee around the points that I don't like about this contour, I can just select them. They become a filled white circle when they are selected. If they are just part of the contour that I'm working with, they are an open square at that time. So I want to delete the filled squares. So I'm going to hit delete, and the contour automatically updates for me with my new points removed. And I can do that in Where Is mode all the way across the data, so I don't have to go navigating around. So I can use this to clean up some aspects of the tracing that need adjustment that I can see at this macro level like this. And then of course you can continue to fine-tune the editing at a desired magnification that is appropriate for what you're going to do. This looks pretty good. So the purpose of what I'm doing right now, this red contour, is to provide a region for the detected objects to run to identify those cholera toxin labeled cells. So if we exit edit mode and get out of where is mode, let's zoom in on a particular section to see what the labeled cells look like. And so you can see here that these cholera toxin labeled cells are these black objects here. And so Mike's going to show us how to run the detection.
So now we really quickly traced our sections, which are in this case going to be regions of interest for the next step. And what we want to do is detect objects, but this time mark them rather than outline them. You choose your marker. I'm going to change zoom levels a little bit so these cells are a little bit bigger in my field of view. We go to the image menu, automatic object detection, and this time we want mark detected objects. Now, this dialog looks an awful lot like the outline detected objects dialog because it is. It's essentially the same underneath, but we're just marking our detections differently. So we have a preset saved for this that we found works good for these labeled cells, called the cholera toxin. And now we're in a preview mode here, so we've just placed some temporary objects that show you what's going to happen when you really run the detection and place actual markers into your model. So now we don't want to use the full image. We're going to use existing ROIs.
What's ROI stand for?
There are regions of interest that we described by outlining our serial sections in the previous steps.
Oh, I see.
So we select them by clicking on them. Tell the program we're done selecting regions of interest, and then say mark my objects. Now, this detection is going to take longer to run than the previous detection where we were outlining because we're operating at a much higher resolution. So there are far more pixels that we need to process for this particular detection.
Well, I was really impressed at how well it was able to discriminate between the purple and the black. I think even though those are both dark colors, it's able to clearly find those cholera toxin cells because they're pure black.
It really helps when the objects that you're looking for stand out from the background with a decent amount of contrast.
So if you're using an antibody stain, you could add nickel ammonium sulfate to your DAB reaction to make it black. I bet that would
certainly help.
Yeah, I think that's a good thing to remember. All right, so the detection completed, and you can see that we now have individual markers over the cell bodies and the axons that were in our field of view. So let's take a quick look to see where else we found labeled cells. So I'm going to pop into where is mode again because it just briefly hides the image data, and you can now see where these cells are, where the markers were placed. So we've got all of the contours, and you can see where the detection ran. So now, if we right-click and select Switch to Go To, I can move in to a new region and take a look at the detection here. And so you can see here that this is a very straightforward way to survey your tissue. It's one thing to know where to look and go look in those specific areas, but if you are doing a brain mapping experiment where you don't necessarily know where all of these individual components are going to be located, this is a great way to get an idea of where those cells actually end up. And so you might be surprised, for example, to see some cells in this area here. You'd consult your atlas, determine what anatomical region this is, and then you can delineate that region.
So Sue, I'm not a scientist. How long do you think it would have taken to do this all by hand?
Well, even with our auto move box, which allows you to trace and automatically move the image or stage as you are working, that increases throughput a fair amount. But I would say that it would take, to do these 8 sections, I would say at least a number of hours.
Wow.
And we did it in just a few seconds. So I think this is a great thing. And you can always go back through and review the results and add in any particular markers for any cells that were missed or delete any that you don't agree with. You can run the detection multiple times also using different criteria such as size or different colors too.
Wonderful.
So now that we have this data, as we said, we've got 3 individual slide images that have each 8 sections. And so now you can see that we've got a contour and some cell markings for these sections on this particular image for this series of sections. But if we were to try to view these in 3D, it's not straightforward to do. So now we want to be able to associate these particular sections with a Z position too, so that we can align them and look at them in 3 dimensions. And so I'm going to switch data files so that we have a little bit easier time manipulating the data. And I'm just going to bring up oops. Yep. I forgot to close the currently open image, so let me do that again. Okay, so now let me show you how to add sections to serial sections. So in order to do that, we've got our individual sections delineated, and you can see that there are markers placed anywhere where the automatic object detection created a blob of the predetermined size for the particular color. And so you can determine whether or not you want these axons included in your markers, and if not, you can delete them. But we're going to start the serial section manager, and I loaded the wrong file. Let me load the right file. And so now we need to start the serial section manager. So I'm going to click on serial section setup. We'll specify the number of sections, the evaluation interval, and the section cut thickness.
Why would you want an evaluation interval higher than 1?
Oh, well, that means the evaluation interval means that these sections are not sequential. They are at an interval separated by every 3 in this case. So every 3rd section is was mounted on this slide for this particular experiment. And so you might have a series that was stained for one particular antibody, one for this particular experiment, and a 3rd series showing something else.
Oh, that makes sense.
And so the cut section thickness is what the microtome was set at when you produced these sections in the first place. And so now you have these, the serial section manager is set up, and I know that these sections are organized from bottom right to top left. So it goes 1, 2, 3, 4, 5, 6, 7, 8. And to add these individual contours to these to each serial section, I'm going to use my handy Where Is mode again and Edit mode. And so I'll select my section, right click, and say Place Contour into Section. and that is section 1. And I'll do it again for each successive section. Until you have all sections assigned to their particular.
So really right now we're showing the program how to add the third dimension to our model.
Yeah, exactly. Okay. Now the last one has been specified, and now we can get out of edit mode and get out of where is mode. And so now, in version 11, we have a new feature that makes visualization of image data in serial sections a little bit easier. What I'd like to show you is now that the serial sections are specified, when I'm viewing a particular section, so remember this upper left section is my last serial section. If I go to display current section only that contour and that image data will be available in the viewing window. And as I move through the serial section manager, it automatically updates to bring me to the next section, so section 19 in this case, and show me the same relative location. So if I were to zoom in even more and switch sections, you can see that the view is kept constant as I toggle between the sections. This is great if you want to then follow a particular cell population through the various serial sections.
So you mentioned earlier that people typically use multiple monitors for this type of work. And so the serial section manager you may have on another window while you're trying to inspect your tissue on the current screen right in front of you. You can use the up and down arrows as hotkeys to toggle between sections so you don't have to take your eyes off of the tissue while you're working.
Right. So now we have our sections assigned to a serial section, but they're still not registered and aligned. So these contours are not on top of each other yet. And there's a few ways that you can do this. But in version 11, we have a new hotkey that is pretty impressive. So you can see in the macro view that we are able to see every section. And now, if we do Control F, all of those sections from the other 8 or the other 7 sections, those contours are then centered on the first section's contour. And so they're already snapped to a registration based on the midpoint of each individual contour across the serial sections. So in one mouse click, I can toggle between a traditional view relative to where the image data is actually located and an aligned view, which would be helpful for us to take this data to a 3D visualization method. This hotkey to quickly snap the contours to the registration of section 1 is available in version 11.
That will certainly make people's lives easier.
Well, yeah. So now what you could do is if you you can continually add to these data files. So as you're working with these data files, you can continue to trace additional regions of interest to build your model with more and more detail and more accuracy. And then keep it registered with the image data that's underneath that you're building this from. Save that data file, and then quickly register and align them, and save that as an aligned data file that you can use for visualization purposes. Since these sections here are very close together in Z, so it's every third section, so they're only 90 microns apart, we're not going to demonstrate 3D visualization with this particular demo. We're going to do a demo of that for each a little bit later on.
Okay.
So to summarize what we just walked through with this particular demonstration, we created a model using virtual slides in which the image contained multiple sections. So this was acquired with a slide scanner. The visual circuit to be mapped was visualized using cholera toxin, so that's how we identified the cells that we were interested in knowing the location of. So you want, when you're doing a brain mapping experiment like this, you want something that labels a specific population of cells. So in many cases, a ubiquitous marker like NuN or Golgi may not be appropriate for the types of models that you're looking to create here. If you're looking for anatomical mapping, Golgi or a ubiquitous marker would be perfectly acceptable. I showed you how to organize those sections according to the rostral caudal order using Serial Section Manager and a combination of where is and edit mode. And in version 11, you can use Control F to register the tracing data according to the center of each section. We viewed and inspected the data, and we navigated between those sections using the Serial Section Manager. So let's move on to the second demo. So the second demo covers a different type of situation in which we have one section per image. And so this is also a virtual image, but only one section's data is present on that particular image. We'll walk through the process of organizing sections according to the rostral caudal order, and then a couple of tips on how to register and align those sections.
So these images probably weren't acquired with a slide scanner.
Actually, in this case, they actually were.
Oh.
This is using the new slide scanner from MBF. And so let's prepare the workspace. So I'm going to clear out the data file and open an image to show you what I'm talking about. Okay. And so here you can see in this virtual slide, only one section is present. And so now we can build a serial section model with this type of data as well. So let's show people how to contour the perimeter of the section.
So I'll show you another way that's available to outline your sections. And this is the tool that I mentioned earlier. It can be used in live mode as well. I'm going to change my zoom so this will look a little bit more like maybe we were on a stage. Let's try this. So this tool is always active, but I want to show you the interface to it, which you can find by right-clicking on your mouse. And when the menu comes up, select Automatic Contouring. So like the object detection methods, there are a variety of parameters you can adjust until you get things working the way that you like. And you can save those parameters that are going to be different for different types of tissue and stains and so on. And we have one saved here in Webinar. Now you get the contouring started by clicking a couple points, and what this does is it shows the software the border that you're looking to follow.
And so by border you mean a high contrast region? Right,
so in this case the border is the outline of this tissue section.
Okay.
Now you can start the auto contouring by a middle mouse click. You can start, stop with a middle mouse, or you can use these buttons here. This button says go, this button means stop, and this button will back you up, and it will back you up a variable distance depending on what you place here in this delete end points box. So if you want to back up big jumps whenever you do that, you would pick a bigger number of points. You can also adjust how fast or slow the auto contour grows, depending on what you're trying to do. So if you're tracing difficult stuff on a stage, you may want to slow things down a little bit. I'm going to start by slowing things down, even though we're not on a stage. So I'm going to say start. Now you saw back there, let me pause this. You saw it back here, the contour kind of veered to the right for a second. Sensed that it had been lost, backed itself up automatically and started searching for the border again. It found it and now it's going again. So if you notice that your auto contour strays while it's going and doesn't stop, you can stop it yourself, back it up, and then interactively get it going again by clicking on the border a couple times. So let's start again. Keep it going slow since we seem to have a slow video feed. We're following the outline. So this is really, now as you see the image shifting as we cross the dotted auto move border, that's when your stage would be automatically moved for you.
That's cool.
It really is kind of neat. So we're coming up on what looks like might be a hard spot. Oh, and we made it around. Now we automatically close to the contour, which is a parameter you can define down here. You can tell the program once the growing side of my contour gets within n pixels of where it started, automatically close it so I'll have a closed contour in my tracing model. And that's how the automatic contouring works.
So we loaded each individual image into Neurolucida and ran auto contouring and saved a data file for each image. And so now we have the situation where we have a data file for each section that we've examined so far. And there are many cases where you also might be working with that similar situation. So we're going to show you how to load those files into serial sections. So I'll clear this out. And now we're going to use an option called File, Open Files into Serial Sections. And so this is a streamlined way to specify these files need to be organized together into serial sections. And so I hold down the Shift key in order to select all of the sections that I want included, all the individual files. And then I'll hit Open, select Open. You can adjust the order, which is in numerical order. And you can see already that it quickly pre-populated the macro view with all the individual contours that I've created for those 10 individual files that I'm now going to merge into a single file. So now it tells me that there are 10 sections. My evaluation interval for this example is 4, and the section cut thickness is 40. And now when I hit OK, you'll see the serial section manager over here populate with all of that data. Okay, so let's zoom out a little bit so you can see how it's generated. And so now we've got all of these contours and all of these images loaded, and you can see all of the individual serial sections that have been generated in the serial section manager. And all of the individual images are loaded in the image organizer. So if I go to Display Current Section Only, which is an icon in the serial section manager, it will show me only the data, both tracing and image data, that is present within Section 1. So all of the data that has a section Z of 0. And so now, as we move between these different sections, you can see the view update appropriately. And so if we were, again, zoomed in a particular region, now when we move through the section, or the serial sections, you can see that we're brought to the same relative location regardless of what image and what section we're looking at. So that registration is a really handy tool. While we're here, I want to show you another new feature for version 11 that sort of makes editing quite a bit easier. So let's say that you are happy with your reconstruction, you love it all except for aqua is not the color that you want to use for a publication. And so previously it can be a little tedious to go through and change the color of each individual aqua contour. And so in version 11 we now have the option to, when you select a contour, in your right-click menu, there's a new option. It's called Select Everything This Color. That allows you to automatically select all contours or markers of that color, and now you can manipulate them all together. And so if we wanted to change the color to royal blue, we could do that.
Well, that's a lot easier than having to go through and click on each one individually.
Oh, yeah, especially when they're not so nicely organized. If you've got aqua sort of all over the place, you spend a lot of time trying to hunt and peck for those individual options. The other thing that's new in version 11 is this option to set contour density. And so I'm just going to apply a crosshatch to every one of these blue contours. So if you were creating an image for publication, you might want to specify, for example, that it is the right striatum that you injected with your drug of choice, for example. And so now, if we go into Display Current Section Only, you can see that the right striatum is crosshatched while the left striatum is not. And so that can be a handy feature.
Very slick.
Yeah. Okay, so one last thing to demonstrate. When I loaded these files into serial sections, they just actually, the alignment is actually pretty good. So this registration that I would have to do is actually pretty minor. But if you're tracing live from under the microscope, or you have a situation where these sections are more diversely dispersed, manipulating them to register and align them can be a complex task. And so in version 11 we have a few new features that make that quite a bit easier to do. So I'm going to pull up the same data that we're just looking at, but I've jumbled them up so they're no longer in such nice alignment. And so you can see in the macro view that these images and the associated tracings are sort of all over the place right now. And so register and aligning these sections so that we can look at them in 3 dimensions using the 3D visualization tool is not going to give us a result that we're happy with right now. And so to show you an easy way to do this, when your data is associated with individual sections, you can display current section only, and the flanking section. And let me zoom in just a little bit. And then there we go. It was a refresh error. Will that be fixed?
You betcha.
Okay, good.
That's why we're still testing.
That's right. So here is section 1 and section 1's image, and I have display flanking sections selected in the serial section manager. And so now you can see that the tracing that is also present is for section 5. And so now, if I select section 5, I can select Move Images and Tracing and move the tracing to the same location over where Section 1 is. And now I can repeat that process for Section 9 and Section 13 and Section 17. And so if you've ever had to do this for your own previous experiment, you know that this can be a bit of a chore, and this is just super straightforward. And so all I'm doing is selecting each section and moving the image and tracing to the same representative location as the previous section. And so we're bringing it all into alignment right now.
That's surprisingly fast.
Yeah, it's very straightforward and easy. And so due to my zoom level and how things were all jumbled about, you can see that I had to do some quick moves in order to see it all together. But in less than a minute, I was able to take that disordered serial section file and bring them into close order. In addition, you could also use Control F to snap those contours and then just move the images using Move Image if you wanted. And so that might be an even faster way to go. And so to sum up what we talked about with this demonstration, when we were creating models using a single section per image, we used auto contouring, which is a right-click option, in order to trace the boundary of your region automatically. We organized our sections according to rostral caudal order by opening those files into serial sections. And to register and align the sections, we used Move Image and Tracing while simultaneously using the Serial Section Manager. Now, Move Image and Tracing is a fine option for getting that gross alignment, as I showed you in the demonstration, very quickly to be correct. You can then change your zoom level and do fine-tune alignment. And if you need to rotate your tracing, that's an option that you can use as well.
So now you've done all this work to create your 3D model, but you're only looking at it in 2D. And as neat as many of the tools we've been showing you are, you'd really like to look at your model that you put all this work into in a more meaningful way. And you can do that with the 3D visualization module. So with that, you can do things like rotate, pan, zoom, set the model into an auto-rotate if you want to make movies with it, and there are many visualization options that you can use to change its appearance.
There's actually too much to even discuss today, and because of that, we have decided to do another webinar specifically on 3D visualization and preparing images for publication. So that'll be a little bit later in the year.
Looking forward to that one.
Sure. Okay, so let's go to Neurolucida.
Okay, we have a previously made model saved in another file, so we'll get out of here. We'll go find our example model. Turn off Display Current Section Only so you can see all your tracing. And you can see by looking at the macro view that there are a number of sections outlined. There's a number of markers placed in here representing different things. And we want to, this is neat and everything, but we really want to be able to look at it as it would be represented in the real world, as a cartoon. So here is the 3D visualization tool. You click this toolbar button and it will load the model in. I'm going to change our markers so they'll be more easily visible. Now, there are options you can see here. We've detected that this data file is comprised of contours and markers. There are no trees. But so let's go into our marker options, We want to make those look like spheres because they're a little more aesthetically pleasing than simple points. We'll go to our contours, and we don't want to look at just the wires here. We want to see a skin on our model. It makes it easier to see the representation we're trying to convey. We can turn off our traced contours. Now you can take different angles to look at your model that you've built. You can do things like adjust the transparency of everything in the model. You can see now how you have a see-through skin. You can also do just the surface skin and change the transparency of that. So now you get a crystal clear view through here, and you can see all your markers in the different sections.
So you get the context of the section, but you are able to see clearly the data that you're trying to represent with this model, so the location of those cells of interest and the various white matter tracks that have been traced.
It's just so much easier to see what's going on rather than a pile of contours and simple markers. So another thing that you can do is set your model into motion, and so it'll just start spinning here. You could do things like create a movie file out of this. It's very smooth on our screen, but since we have the video lag, it may be a little bit jerky on the other end of the webinar, but trust me, it's very smooth in action. Now you can also do things like change the angle of the rotation with this little tool here, and you'll see as I rotate it, the axis of rotation changes angles, and you can get different views depending on what you're trying to see. So really this takes all your work that you've done up to now and puts it into a nutshell that you can get your take-home.
I like that auto-rotate because I know that I'm not very smooth when I use the mouse, and if I wanted to create a movie, it would just be too jerky, and I'm not willing to cut down on my caffeine intake, so I like that feature.
So that's the 3D visualization in a nutshell.
Okay, well, cool. So we've covered a lot of ground today. We've talked explicitly about brain mapping and that technique and how you can use Neurolucida to generate that type of data. As with any experiment, the details and the level of accuracy that you require in order to prove your hypothesis is determined by you, and so you would create the model with that needed amount of accuracy. But some tips that we have for efficient brain mapping include configuring your workspace. We showed you at the very beginning how to utilize custom toolbars and toolbar buttons in order to organize your analysis tools for the particular application that you're using. And I organized all of that by creating a profile specifically for brain mapping. We created serial sections a number of different ways, and that allows us to create the data and visualize it not only in the original location as they appear on the glass slide, but also relative to how they existed in the brain itself. With Image Organizer coupled to the Serial Section Manager, you can navigate between sections very easily, and when you do so, it'll show the same relative location as you move between sections. This makes it very easy to evaluate your data. And finally, we talked about a various number of automated tools that can help speed your data collection. Whether you're using automatic object detection to help you outline your regions, to quickly run a detection to look for your labeled cells, or use automated contouring in order to more closely delineate your region of interest. You can combine those with other manual methods at will for your specific application to help improve your throughput and efficiency.
Now, there's a lot of tools that we went through today that really come together nicely.
Yeah. And so many of those tools all of those tools that we talked about today are available in previous versions of Neurolucida. And we took pains today to talk about how version 11 is just improving the throughput and efficiency. While we talked about creating all of this data today using virtual slides, you can create this data using your slides under the microscope and just trace as you go. So that option is available to you. One other thing that I want to make sure to just highlight is actually the topic of an upcoming webinar, which is Biolucida Cloud. That's a new product from MBF Bioscience, which allows you to share image data with your colleagues by putting the image data
just in one central location.
And where would that be?
Wherever you have your Biolucida Cloud server and set up.
And so you can create collections and share those with people you are collaborating with, or bring those images down into Neurolucida and continue to collect data from them.
Yeah, you could have multiple people looking at the model like we were just now from different locations.
So if your server could be right next to you, so a local area
Sure.
network, or
Sure.
in Chicago.
Sure.
Okay. So that's going to be a topic of another webinar coming up shortly, I think in April. So stay tuned for that. So in summary, with Neurolucida, you can work directly from the microscope or with images. Using the input, you can reconstruct anatomical regions from serial sections and map the location of cell populations. You can visualize those models in 3 dimensions using the 3D visualization tool. And you can always quantitatively analyze your data by opening those files in Neurolucida Explorer,
Great.
which we didn't have time to get to today, but that is available to you. So we want to thank you for over 20 years of excellent feedback. We also want to thank the NIH for numerous small business innovation research grants that allow us to really push the envelope on new and exciting advancements for analytical research. So contact us at info@mbfbioscience.com or give us a call. We can set you up with a free trial of Neurolucida. Or you can go to our YouTube channel and view some videos of various features in action. Thanks for your attention. We'll see you next time.
Thank you.